Thank you for the invitation to be here. It's a real honor to be here in the Pieter Cullis Invitational Lecture. I know you don't like the memorial aspect of this, but I'm very much honored to be part of the series, and look forward to presenting. And we had a conversation right before where I told my talk be very fundamental. So unlike many of the other talks that you might hear, I really will walk you step by step through DNA nanostructure synthesis and an assembly. And we are exploring applications, but nowhere near lipid nanoparticles for the applications that we are exploring. Okay, so as I said, our research group takes the molecule DNA out of its biological context, and we use it as a building block to make nanostructures. And then we take these nanostructures and we apply them to problems in both biology and material science. And so what I'm going to do is I'm going to start by giving you a short motivation for the usefulness of DNA nanostructures in the field of drug delivery. Now, as we saw this year with the COVID-19 mRNA vaccine, witness medicine, right? Scientists first developed small molecules as drugs, and then about in the 1980s, we saw the development of proteins and peptides as drugs, like antibodies, and now we are seeing the emergence of a new class of molecules based on RNA, based on DNA, based on nucleic acids, and in my opinion, they are going to make a very big difference in medicine. And to explain this a little bit, many of you may know this already. So if you think about small molecule drugs, they are meant to target proteins, right? And they're meant to inhibit mechanisms that proteins might be using for disease. Now, if we go one step backwards to the mRNA gave rise to the protein, and we think of targeting that mRNA, then the drug design aspect of this is going to be highly simplified. So while we have 20 amino acids and a very complex protein that folds into complex shapes, here what we can do to basically the translation of mRNA into proteins, we can actually design an RNA therapeutic that has a complementary sequence to this mRNA. And as a result, through various mechanisms, we can actually silence this gene and stop the formation of this protein in the first place. And this is really important for multiple diseases, including cancer, where many of the cancer driver proteins are considered undruggable. That is, it's difficult to find molecules that can actually bind to one of their pockets to inhibit their action. So DNA and RNA therapeutics, of course, you've seen them at work as mRNA therapies in vaccines, and this is a second application of DNA and RNA therapeutics, namely gene silencing. Now, why haven't we seen a lot of these in the clinic already? Just to say here, I forgot to tell you that if you don't know, there are now siRNA or gene silencing RNAs in the clinic, including using lipid nanoparticles pioneered by Pieter Cullis. And so there are some, and many of them are in the pipeline, but this is not a very general strategy. And the problem with DNA and RNA therapeutics is that they have delivery issues into the body. So basically, they're not very stable biologically. They're sensitive to nucleases in our blood and multiple other biological barriers. If you are able to get them close to a cell, they have poor cellular penetration, as you might imagine, being large macromolecules that are negatively charged. Some of them cause immune responses, and naked DNA RNA is going to have a very fast clearance in vivo, and is going to basically clear mainly localized in liver if it can actually last that long. Now, one might imagine that you can create nanomaterials, nanoparticles that might encapsulate nucleic acid therapies and deliver them to the site of action. And indeed, you saw that happening in really something really historic that happened, and that is actually Canadian-based, right? We saw lipid-based nanomedicines that encapsulate mRNA vaccines and have actually basically saved millions of lives against the COVID-19 disease, and have shown us basically how powerful these strategies are for vaccines. While lipid nanoparticles are a very mature technology, they do suffer from some limitations, and I'm listing some of them here. And I really want to go from the specific to the more general here. They are systemically. Typically, they will mainly deliver to the liver, and that's in fact the basis for an siRNA therapeutic on Patro, which is used to packaged into a lipid nanoparticle and used to deliver to the liver, basically to cure liver disease. Once lipid nanoparticles, or many of the other modalities, I'm not just going after lipid nanoparticles here, many of the other modalities as well, once they go into cells, they allow only a small fraction of the nucleic acid to be in the cell cytosol where it should exercise its action. Mostly the nucleic acid is trapped in endosomes. And really what's the most important thing in my opinion is that we still don't have in drug delivery, and with these nanocarriers, we still don't have this predictability. That is, we don't know what composition, size, shape, etc. of a particle is going to give us, for example, delivery to a specific organ or to a specific cell within that organ. So we still don't know how to rationally design nanomaterials in order to achieve targeted delivery to specific cells. And in parallel to this, it has emerged in drug delivery that it's not just the chemical vehicle that matters. The size and the shape and the way molecules are presented on the surface of a drug delivery vehicle determine pretty much everything in its biological distribution and activity. Essentially, how fast it is cleared by our immune system, what organ does it target, whether it enters cells or whether it enters, whether it basically goes inside a cancer cell, etc. All of these features depend on the size and the shape and the molecular presentation. Yet most of the nanomaterials that are approved for drug delivery right now are spheres, when we know that many other shapes might be superior for some applications to spheres in terms of drug delivery. And the precise control of size, shape, presentation of molecules in an asymmetric or asymmetric way on these structures is somewhat difficult right now for most of the nanomaterials. So if we wanted to acquire the systematic knowledge or fundamental understanding of the relationship between the size and the shape of a nanomaterial and where it goes to which organ it targets or to which cell it goes to, if we wanted to do this, we might borrow something from medicinal chemistry. We might actually develop what we call structure-activity relationships. That is, take the same material and make a very large number of big library of different sizes and different shapes, and basically correlate each size, each shape with where it's delivering, what cells is it targeting, and whether it is, for example, silencing in those cells, gene silencing, or doing any other therapy in those cells. However, the problem is, in nanotechnology, there aren't many materials that you can use, a single material that can be used to create a very large variety of sizes and shapes and composition and presentations, essentially. And so as a result, when you go to the literature and you read about the size and shape dependence of drug delivery, you find a lot of contradictory things. In some cases, for example, rods deliver better than spheres, and for other materials, it's spheres that deliver better than rods, etc. And that is because those studies are done on different materials. There isn't really, as I said, one single material that you can use to create these very, very large libraries and systematically vary size and shape. And that would be very, very important to obtain a fundamental understanding of the biological activity of a nanomaterial. So our research group, as I said, is interested in using DNA as a building block. And what we would like to do is package DNA or RNA into nanostructures. And we're going to use these nanostructures as drug delivery vehicles or as nucleic acid delivery vehicles. Now, as I'm hoping to show you today, unlike many of the other nanomaterials, DNA nanostructures can be made with deliberate variation of size, geometry, stiffness, flexibility, and they are made as monodisperse materials with a single molecular weight. They can be used to position molecules in any kind of presentation that we need in order to target specific receptors on cells. So very, very precise size and shape control is what I'm hoping to show you today. Now, our lab is a little bit different from other labs in DNA nanotechnology in that we don't just use DNA base pairing in order to create our structures. What we do is we combine DNA base pairing with a lot of concepts in organic, inorganic chemistry, supramolecular chemistry, polymer chemistry, and we create these hybrid materials that expand the applications and the function of DNA nanostructures without making them from a very large number of constituents. And so, as opposed to maybe you might have seen before some approaches in DNA origami, such as in DNA nanotechnology, such as DNA origami, where you can actually create bunny rabbits and the Mona Lisa and all of these very, very complex shapes from DNA. These structures use hundreds of different DNA sequences in order to create one of these structures, 200, 300. And so while they're very, very interesting molecules, they might be difficult to be applied in drug delivery applications because of their complexity. In fact, you would need to know the biological activity of every one of the components of these drug delivery or of these DNA origami structures. So instead, what we do is we use DNA minimal structures made from a minimum number of DNA strands and decorated or incorporated with other molecules such as organic, inorganic, and polymer molecules to diversify their applications. And so I'm going to walk you through how we make our structures. And I'm going to start by talking to you about DNA cages. What we're going to do is together walk through the synthesis of a DNA cage step by step. Okay. So over the years, and I just wanted to say also the reason why I'm going through fundamentals. When I started in this field, the field was really just in its infancy. There was Ned Sleiman and one or two more others Ned Sleiman is a pioneer of the field, but we were very few working in this field. So we had to kind of invent all of these tools and technologies ourselves. We did not have the luxury of using other people's tools and technologies. And that's why we kind of went step by step and put together our structures ourselves. Okay, so how do we make a DNA cube? And I'm going to show you how to make a DNA cube from 4 DNA strands, right? Not 200, just 4. Okay, so the idea is the following. What we're going to do is we're going to start with this brown DNA strand shown right here. And by the way, I cannot see my audience at all, so please free to interrupt me if something is weird in my presentation or you're not seeing something, because I don't see the feedback from my audience. Okay, so we start from our first strand shown right here. And we can, of course, make DNA strands on a synthesizer, which we have in the lab, or we can basically design their sequence and order them from a supplier. So what we're going to do is we're going to design this part of the DNA strand and this part of the DNA strand to be complementary to the backside of this DNA strand shown right here. So that when these two come together and form a double helix, this and that close on this green strand. As you can see now, this is cyclized basically onto the green strand. And we can continue to do this. So let's make the green strand here complementary to the backside of this blue strand to cyclize. And then finally, we introduce the fourth strand, this dark blue strand here. Again, same thing, the light blue strand cyclizes on the dark blue strand. And now we're ready to close the cube. So what we're gonna do is basically make this strand here, design this strand here to be complementary to the backside of the first strand shown right here. So that it closes upon itself and creates this cubic shape shown right here. And now if you see it here, you have DNA on the top and DNA on the bottom. In fact, you have 8 positions, 1, 2, 3, 4 on top, 4 on the bottom. They can be of different DNA sequences. And so by hybridizing these positions with DNA strands, and if these DNA strands are carrying whatever we want, drugs, nucleic acids, polymers, whatever it is that you want to decorate this structure with, then we will be able to place them exactly where we want them to go. So let's say, for example, ligands for specific receptors, they'll be able to be placed exactly where you want them to go. So this is shown schematically here, where the strands are basically closing upon each other, and then the whole thing is closing upon itself because of complementarity. And as I said to you, 8 different strands can be hybridized on the top and on the bottom if these strands are carrying on them a group, whatever group it's going to be patterned on the prismatic structure. And just like we can make these cubes, we can also make a triangular prism, for example. It's very simple. Just stop at the 3rd strand here and make this part here complementary to this part. We can make pentagonal prism. We don't stop here. We continue with a 5th one, and we make the 5th one complementary to the 1st one. And these are all easily synthesized right now. So any of you here can actually order these strands from a supplier. They're very cheap right now. And then you can put them in a buffer solution with a little bit of salt added, heat them to break all the hydrogen bonds, cool them back down, and you obtain the cube or whatever structure that you're trying to form in 100% yield. So this is a very, very simple and efficient strategy to create all sorts of size and shapes from DNA structures. All right, so we can create different shapes and sizes, but one thing as well that DNA nanostructures have that is really unique to them is that they are what we call molecule responsive. So if you imagine in a disease environment, let's say in a cancer environment, there are typically molecules called biomarkers that are either overexpressed in a disease environment or only expressed in the disease environment. And what we can see here, what we can do here is we can actually design DNA cages that encapsulate their drug cargo on the inside and stay closed. And when they go to the disease environment and when they encounter only this biomarker, only then will they open the cargo. Molecule responsive conditional release of the cargo. This is something that you can achieve with DNA nanostructures because of how responsive they are, and you cannot really very easily with other materials. And so I'm going to show you first a very early example that's really non-biological, but just to show you the strategy. And then I'm going to show you more biologically relevant examples. So a long time ago, we designed this nanotube. So it's basically an elongated kind of DNA cage. And we designed it to encapsulate on the inside gold nanoparticles. The gold nanoparticles are trapped on the inside. Okay, and what we're going to do is we're going to add a DNA strand. This is basically representing, if you want, the biomarker that in the future for biologically relevant application we will use, but right now it's just an added DNA strand. So this DNA strand, here's what it's going to do. This DNA strand is going to come in and is going to remove 2 of the strands here that make these cages double-stranded. These cages then become single-stranded as a result of single strand, the gold particle slips right out. It's not as much attracted to the scaffold, and it slips right out and it is delivered. And what does this behavior depend on? Let me tell you about one strategy that people in DNA nanotechnology use a lot, and that is strand displacement. And this forms the basis of many of the nanomachines used in DNA nanotechnology, and it is going to form the basis of our releasing or conditional release strategies. So here's the idea. The idea is, if I have a double-stranded DNA shown right here, where this blue strand is hybridized to this strand here, it is complementary, but this strand has a short, what we call overhang, single-stranded part right here that is just a little bit longer than the blue strand right here. Now, these two can form a very stable double-stranded DNA. But if you add a fully complementary DNA strand, A prime, basically, what happens is A prime is going to come in and it's going to basically grab this overhang here, the single-stranded part, and it's going to kick out this blue strand shown right here, okay? So what happens is, and if you can imagine, now we have a more thermodynamically stable double-stranded structure with more DNA base pairs, okay? And this reaction, it turns out, is really fast. It occurs in seconds. And the reverse of this reaction is very difficult because the short strand does not find an overhang or a single-stranded DNA in order to grab it and go backwards. And so this is the basis of a lot of, as I said, nanomachines with DNA. We're going to use it in our case. So what we're going to do basically is we're going to have our DNA strand here complementary or fully complementary to these strands, and that's how it's going to remove them. Oh, so it's shown right here. You can see here, those strands are longer than the cage, and the fully complementary DNA strand grabs them and basically kicks them out. The cage becomes single-stranded and the gold particles slip right out of there. And we verified this using both TEM, basically microscopy, and multiple other spectroscopy techniques. Okay, so based on this, of course, let me show you now a little bit more of a biologically relevant strategy. So what we have here is an unzipping cube, if you want, and this is to stay closed. This cube was designed by Catherine Bujold, who's now herself a professor at McMaster University. So Catherine designed this cube to stay closed, and it's only going to open in response to an RNA that is present in prostate cancer cells, but it's not present in normal cells. If you want to know further, this is actually a gene fusion product, and it is only in prostate cancer cells and not in normal cells. So what we're going to do is we're going to design this cage to stay closed and only to open in response to this prostate cancer cell specific molecule. And the idea is shown in, hopefully, if I can, yes. So you need basically two of these strands that will release, or that will do strand displacement and release two sides of that cube, and the cube completely flays open, and then if there's on the inside a cargo, it will be either revealed or released. And in a second step, what Catherine did is she designed what she called a suitcase prism. So it's like a suitcase, and on the inside, it is carrying a gene silencing a strand, so an siRNA strand that is sitting on the inside, and the cage is completely inactive in cells if basically it is in cells that don't have the trigger. But if it goes in cells that carry specific microRNAs, these microRNAs again are going to cause strand displacement of these two sides right here, kicking out the siRNA, at which point this siRNA can undergo gene silencing and can basically do whatever action that it needs to do. So conditional drug delivery vehicles using DNA nanostructures are possible, and may find nice applications actually for very targeted drug delivery. Okay. After this short introduction into DNA cages, I'm going to move and introduce to you another class of DNA structures that we're also very excited about. And this is a class of structures that combine polymers with DNA. And I'll tell you in a second why we want to do that. All right, DNA as a language. You have basically adenine binds to thymine, guanine binds to cytosine. So it's a language made of 4 letters only. And in fact, it's because the language is so that base pairing is so predictable and that you can actually generate these really highly controlled DNA nanostructures that we know about. But of course, this is a very limited alphabet, okay? On the other hand, polymers have a very, very, very large set of interactions that they can introduce. So I list some of these right here: hydrogen bonding, pi stacking, fluorophilic interactions, the metal coordination, et cetera, et cetera. By doing so, what they introduce is the ability to make very large extended structures with a very small number of components. Okay, so one single component, you know, that might be amphiphilic can actually very organized structures. And so combining this ability of, and the various languages, if you want, of polymers with the very limited alphabet of DNA is advantageous, because what we can then do is create complex structures without having to use hundreds and hundreds of different DNA strands. And so this is our motivation, but we're going to go one step further here. So normally, conventional polymers, as you might know them, are polymers that look like this basically row of penguins in Prague, in the Czech Republic. All the penguins are identical to one another, except for the little, I don't know if you can see them here. There are some pigeons sitting on top of each of the heads of the penguins. So normal conventional polymers, they're made by taking one monomer and basically repeating it down the chain, okay? You can make blockopolymers, graft polymers, but essentially conventional polymerization gives you that kind of polymer. On the other hand, natural systems actually use a very different strategy to make their molecules, right? Natural systems use macromolecules that are what we call sequence controlled, right? So DNA, RNA, peptides, proteins, even some sugars on cells are sequence defined. And that's because the information contained in a sequence can actually give rise to structure and to function using a very small number of starting building blocks, right? So how do we make sequence-controlled polymers in the lab? It's not that simple. But what we thought is that we do have an instrument in the lab that is capable of making sequence-controlled polymers, namely the DNA synthesizer. Now, the DNA synthesizer is an amazing piece of engineering developed over and the amazing thing about the synthesizer is that, you know, you can come in, as one of my students may come in and fill out the bottles in the synthesizer, go to the computer, type their DNA sequence, and what the synthesizer is going to do is going to add one by one each DNA nucleotide in the sequence that you asked it to do it. And the coupling yield in each of these additions is 99.9, sometimes even 99.99% at each of the steps. This is how good the coupling steps are. So our DNA synthesizer is capable of making specific sequence-controlled polymers, DNA and RNA, okay? But we are chemists, and we can actually repurpose the DNA synthesizer to make synthetic sequence-controlled polymers. Okay, so what we need to do for this, and I'm not going to go over the chemistry here, but what we need to do for this is we need to understand the chemistry that the synthesizer uses. And rather than use nucleotides as the monomers, we are going to use our own artificial monomers inside this exact same chemistry that the synthesizer uses. And so I show you a small subset of the molecules that we have made in the lab. So you have pi stacking, hydrophobic, peg chains, fluorophilic interactions, sugars, amino acids of all sorts that we were able to add. And literally what you do is you basically add them to different bottles on the synthesizer and ask the synthesizer to create these sequence-controlled polymers in any sequence that you want by basically programming them as additional bottles in the synthesizer. And as a result, now you can expand the number of interactions that DNA and RNA strands can engage in by a lot, but just adding a handful of these different unnatural DNA molecules. Okay, I'm going to show you one application of these DNA structures. Just excuse me, I'll just drink some water. So one of the applications of these DNA sequences is in what we're going to call self-assembly. So here's the idea. The idea here is that we're going to create one of these artificial DNA artificial molecules, and the molecule is going to have a long alkyl chain. So a long C12 carbon 12 chain that is in this monomer here with the same chemistry that the synthesizer uses to create its strands. Okay, so then what we're going to do is instruct the DNA synthesizer to make a DNA strand, so a 19 base, let's say, DNA strand, after which we're going to tell the synthesizer to add this C12 chain, this carbon 12 chain, 12 times. Okay, C12, 12, so 12 times. And remember, the synthesizer is actually making DNA molecules, right? So these DNA monomers are punctuated by phosphates, right? So our C12 chains that are being added in the end will be also punctuated by these DNA, by these phosphates. Okay, so now we're going to take this off of the solid support, and what do we obtain? Well, we obtain on the one side the DNA strand itself, which is hydrophilic. And then on the other side, we obtain the C12 chains, which are hydrophobic, okay? And by adding dication and a buffer solution to this, what we will see is the hydrophobic units will want to hide from the solvent and will assemble into these spherical micelles. So this is a single molecule, and hundreds maybe of these molecules, or 50 or of these molecules are going to be assembling into spheres where the inside is made up of the hydrophobic C12 chains and the outside is made up of these DNA strands. Okay, we're going to call these spherical nucleic acids. Really simple. You just obtain them from a single DNA strand that is amphiphilic, so it has a hydrophobic and a hydrophilic structure. And since we created this on a DNA synthesizer and it is monodispersed with one molecular weight, we also make these spherical nucleic acids, they assemble into very low dispersity spherical nucleic acids. So they're really very, very well defined. And of course, we can change the structure of the monomer on the inside. Small changes in the structure of a monomer dramatically change the assembly that you get. So for example, Michael here, instead of using a linear C12 carbon 12 chain, here he used a branched carbon 12 chain. So basically, as you can see here, the phosphates are here, okay? And the carbon chains are coming out as branches from these monomers. Okay, and what Michael showed is that these actually assemble into fibers rather than spheres. So why do they assemble into fibers? We were very intrigued fundamentally on why this was happening. So we teamed up with a group of Jianing Li at the University of Vermont. She has now moved actually to Purdue University. So Jianing does all atom molecular dynamics simulation in her lab. And so what they did is they took our structures and they simulated them. And what they found is, as I told you here, there's the phosphate backbone shown right here, which is hydrophilic, and the alkyl chains are hydrophobic. And upon heating these structures, what happens is the phosphate backbone curls. Okay, so we're going to call these curlomers. It curls and forms this disc shape right here so that the alkyl chains are hidden on the inside and the phosphate is on the outside. And then these discs sit on top of one another. And in the end, they, in order to hide the alkyl chains, and these actually form very nice and very rigid fibers with this mechanism. And so this is what it looks like, with the alkyl chains and the phosphate in here and the DNA strands on the outside. And it's very simple design that you can actually change the morphology. So I don't have time to tell you too much about this, but we are with very simple changes in sequence and in composition of our polymers, we are discovering really a wonderland of different morphologies and different structures. And so the nice thing about these is that they are even more simple than regular DNA nanostructures. So rather than making a cube from 4 strands, now we're just making a fiber from 1 strand. Okay, so they're very, very easy to synthesize and easy to assemble into the final structures. So if you're interested in knowing more, we have published a few papers on those mechanisms and on the effect of every one of the parts of these amphiphilic structures on what morphology we obtain. And so I invite you to read those if you're interested. So the next thing to do is we have our cubes or our nanotubes, so our nanostructures, and we now have this class of molecules that has a DNA and these hydrophobic or other synthetic polymers, sequence-controlled polymers attached to them. What happens if you actually put the two together? Okay, so you take these DNA nanostructures that allow you to position anything and everything exactly where you want it to go, and you position these polymers onto this DNA cage. And what I'm going to do is I'm going to show you one example, and just to show you how sometimes you can obtain really from the assembly of these structures. So this is simply one of the amphiphilic molecules that we attached onto DNA. In this particular case, this amphiphile is more like a dendron. It's dendritic, as you can see, carrying 4 C12 chains, you know, as 4 arms, basically, on a single structure. But essentially, this can be also achieved with the polymers that I showed you before. Okay, so when Tom placed 4 of these DNA strands on the top face of the cube, okay, what he did find is that this cube no longer was there anymore. What happened is he made, in almost quantitative yield, a dimer of 2 cubes. And so if you imagine, the cube has one face that is lipidic, you know, that is hydrophobic, and this other cube also has a face that is hydrophobic, and those faces want to hide from water, and so they undergo a kind of a hydrophobic handshake, if you want, in order to hide the interior lipidic chains shown right here. And so you get really very nice ones, and we found later on that this is actually highly predictable. So if you make these chains longer, you start getting, instead of dimers, you get trimers, and instead of trimers, then you get tetramers, et cetera, et cetera. So you can really control it. So I said to Tom, well, why don't we put 4 on the top and 4 on the bottom? So basically attach the nanotube, the cubes right on top of each other to make basically a chain of cubes. But that is not at all what formed. Instead, what formed is the alkyl chains on the top went inside the cage, and the alkyl chains on the bottom went also inside the cage, and they underwent a intramolecular handshake, where now what we have is inside the cage itself, we have a hydrophobic interior. And this hydrophobic interior, of course, now is capable of encapsulating small molecules, right? Because small molecules, drugs, are typically hydrophobic. And again, in collaboration with Jianing, what Jianing showed is that this kind of collapse of the interior happens very, very quickly in these cubes. It distorts the whole cube, but as it turns out, actually the entire structure is more stable than a cube without those hydrophobic units on the inside. So as I told you, now what we can do is do the same encapsulation and release that I showed you with gold nanoparticles and I showed you with siRNA, but now we can do it with small molecules. So we can encapsulate a small molecule on the inside because it is hydrophobic, and now we have a hydrophobic pocket. And by adding strands that remove those strands carrying the lipid chains, what happens is we lose the lipidic environment on the inside and the molecule is released. I cannot see the chat, but if there's anything that you guys need to tell me, you might want to say it in words, because I can't really click on the chat while I'm in presenter mode. Okay, So this is just an example of what emergent properties, surprising properties, can actually come out of these sort of like protein-like assemblies, where we're placing everything exactly where we want it to go in any pattern, symmetric or asymmetric. So now let's go to some of the biological properties of our molecules. We have now a large library of these structures, and we are ready to start seeing if these structures can overcome some of the problems that are faced by nucleic acid therapies, such as gene silencing therapies. Okay, first question. Are our 3D DNA structures susceptible to nuclease degradation? I told you that there are enzymes in our blood that are capable of degrading DNA and RNA. Obviously, it would be a very wasteful situation if all of our cages here were degraded very quickly in the body. But it's interesting. It's really supramolecular idea. By packaging our structure in these three-dimensional shapes, such as prisms or nanotubes or spheres or fibers, what we're doing is we're hiding many of their ends from their the enzymes, especially exonucleases that would come in and degrade them. And so as a result, we see a very large extension in their lifetime in serum. So just as an example, if you look at the lifetime of one of the strands that make up this prism, it lasts for about 4 hours. But if you actually make 3 of these strands go into a prism, the lifetime now becomes 18 hours. And if you just cap the ends here with very short ethylene glycol chains, now the lifetime is 60 hours. So really a very respectable lifetime. And this before we decorated this cage with anything like polymers, which are of course going to extend the lifetime further. I don't have time to tell you about this, but one of my students, Tyler, has actually developed also another really cool technique to further stabilize these structures. And I'm happy later on to maybe in another occasion to share this with you. Spherical nucleic acids are also extremely stable after engineering them. They can be stable after 6 days. All right, very good. Second question. DNA and RNA are difficult to bring into cells. And again, this is a supramolecular question. By packaging DNA nanostructures into these like globular shapes, sort of spheres of dense negative charges, would there be any receptors on our cell surfaces that might enable or help with their uptake? And the answer is yes. One thing that is very important though here, and I urge you, if you ever are doing any DNA or RNA delivery into cells and monitoring this by fluorescence microscopy or FACS, I urge you to read this paper. This is a paper that is cautionary tale to show you what control experiments you need to do in order to make sure that you're not degrading the DNA and observing degradation products. So if you're very, very careful, you still see uptake of our DNA nanostructures inside cells. The most robust uptake that we've seen are our spherical nucleic acids. They really go very nicely into cells. And recently, Jath in our lab has actually figured out that this uptake is mediated by scavenger receptors and occurs by a caveolin-mediated mechanism. So they go into cells, they utilize a different kind of strategy in order to enter cells. But of course, if they go into cells, that does not necessarily mean that they're actually going to be active, because as they're going into cells, they might be going, most likely be going through an endosomal uptake, and they need to be freed out of endosomes in order for them to act in the cytosol and do, let's say, gene silencing. So the next step that we wanted to determine is, are our structures, do our structures show therapeutic activity? And for this, we teamed up with a colleague of mine at McGill University, Masad Dama. The Dama lab has pioneered a class of molecules called fluoroarabinose nucleic acids, or FANA nucleic acids. These nucleic acids are fantastic in that they undergo, first of all, they're less immunogenic, as gene silencing tools. They're very efficacious, and they undergo gymnosis that is basically, they can go into cells without needing transfection agents. So the question that we asked was very simple. Can we actually take the FANA gene silencing nucleic acid, and can we attach it to our alkyl chains, our C12 chains, thereby forming spherical nucleic acid, where the outside is these therapeutic or these gene silencing nucleic acids, and the inside is these hydrophobic structures. And would this basically silence gene production basically in cells? And the answer to this was to our happy surprise that these are much more potent actually than the nucleic acid itself for gene silencing. So this is an experiment where we're doing gymnosis, essentially transfection-free gene silencing, and we see that there's more silencing actually affected by our spherical nucleic acids than by the FANA molecule by itself. And critically here, this depends on the presence of a cleavable spacer between the nucleic acid therapeutic and the alkyl chains on the inside. And we determined that actually this cleavage is necessary, but it occurs inside the cells. So the whole thing is actually stays as a spherical nucleic acid. It is taken in, and intracellularly endonucleases mediate the cleavage of the therapeutic, which then starts actually acting, and it acts better than the therapeutic by itself. So this is good. And the last thing I wanted to share with you is some of the work on their in vivo behavior. We're really only starting and scratching the surface here. And if any of you, I don't see my audience, or if any of you here have in vivo models that you would like us to collaborate with you on, I would be delighted, because we really would like to understand the in vivo behavior of our structures. They seem to be doing very, very well, but we need to understand it further. Okay, so the first thing that we wanted to ask is, can we use them for drug delivery? In this case, what we're going to do is we're going to ask two questions. First, can we encapsulate small molecule drugs on the inside of our spherical nucleic acids? Will they deliver those small molecules to cells? And secondly, what is the in vivo behavior? What is the biodistribution of these structures? Okay, and for this, we teamed up with Dr. Larry Panache. Larry is actually an oncologist at the Montreal Jewish General Hospital. And among the patients that he treats are patients that have developed leukemia and have developed resistance to chemotherapy. And there is a drug actually developed by Novartis called BKM120. It's a PI3K inhibitor, so it's a kinase inhibitor, and it has been shown to be to chemo. However, this drug has severe side effects, and that's why it's kind of still clinical trials and has not really made it through to the clinic. And one of the big side effects is that this drug crosses the blood-brain barrier and causes depression, which is really not desirable at all for anybody, including, of course, cancer patients. And so the question that we asked was very simple. One, can we encapsulate BKM120 and deliver it to cells using our spherical nucleic acids? And two, what does this biodistribution look like? And can we use the SNAs, the spherical nucleic acids, so that we do not cross the blood-brain barrier, and we essentially do not deliver this drug, this BKM120, to the brain, where it will cause side effects? Very quickly, yes, indeed, we can encapsulate BKM120, and in primary leukemia cells, we see definitely that they act as they should. They resensitize patient cells to chemotherapy. And now let me show you just a little bit an IVIS, basically of a fluorescence image of where they are. So what you're looking at in yellow and in blue and in green are the spherical nucleic acid as it's distributing inside the mouse. And I'm going to stop it here just for a second. I did not tell you that, I did not mention it here, but I wanted to mention here or remind you that what we really don't want is for our nanoparticle to be stuck completely in the liver. Okay, so I'm gonna play the movie just to show you. So the distribution once again is green, blue, yellow, and show you the organs. And what we really want to make sure is that it's not stuck in one of these organs, in one of the organs that normally, like the liver, for example, or the kidneys. And indeed, that's what we see. We see distribution throughout the mouse. We do not have any targeting ligands or anything here. And we see that our nanoparticles do not all concentrate in the liver, which is very nice. I think the next thing is the kidneys. And you will see some of it in the liver and you'll see some of it in the kidneys, but the main thing is that the particle is distributing throughout the animal, right? I think the next thing is the lungs. And you can see some of it right here. You can see it here, right? But basically, again, it does not just sit in the lungs, although that would have been nice too. And importantly, for this particular application, very little of it actually ends up in the brain. And so we can actually use this to deliver, let's say, BKM120 or other drugs that we do not want to have any neurological side effects from. Okay. This is a study in tumor models where we see that they also concentrate in the tumor. And so they are quite promising. What's the most promising right now for us is, of course, in vivo gene silencing. So our first studies with some of our molecules have shown that indeed our particles or our structures are capable of gene silencing beyond the liver. So they can actually silence in the lung, and they can silence in the skin and heart, adrenal gland, and even fat. So we're scratching the surface, as I said before, but we're very excited that these particles are not basically concentrating only in the liver and just doing gene silencing only in the liver. And we're excited to do the next studies to explore this further. So I hope I showed you today through a very fundamental talk that tried to walk you through how we make our structures and what they look like and what they do, that we do have a very large library that we can create out of our nanostructures. DNA can be basically molded into very large numbers of sizes and shapes. And what we can do now is we can actually use these and make very large libraries. And in a high throughput way, we can actually now start studying the biodistribution of these different particles and find out basically what is the relationship between the structure, the presentation of molecules on its exterior, and where it ends up, what tissues and what cells it targets, so that we can develop a true understanding and a predictable way to predict essentially the biological activity of a particle from its structure. And I haven't talked to you about many of the other things that we do, but we're very excited about other strategies that we use, and we're trying to also implement them not only for delivery the way they are, but actually recently we're starting to work with Larry Lanz, who's another medical doctor at one of the Montreal hospitals, in order to incorporate some of our technologies in Pieter's lipid nanoparticles, actually. And we're also very excited about the kinds of things that we can do with our structures when they're put in with a much more mature technology, which is lipid nanoparticles. And it's to thank my wonderful students for their contributions. They are fantastic. I'm very, very excited to be working with such a great team of researchers and people. And I'd like to thank the funders, of course, and you for your attention.
Well, thank you very much, Hanadi Sleiman, for an exciting, profoundly thought-provoking presentation. I think all 50+ individuals on the line have learned a lot today about what's going on the bleeding edge of discovery, and this is really quite opportune that you shared this with us today, because we are looking ahead now to what next in our field. And given the major proof of concept that happened around lipid nanoparticle delivery in the COVID vaccine case, We're obviously open to all kinds of other new exciting developments and opportunities in this rapidly expanding field. So it's my pleasure for the next 15 minutes to entertain questions from our participants, and I'm going to perhaps give Dr. Pieter Cullis the opportunity to ask the first question or to make the first comment. So Dr. Cullis.
Yeah, Hanadi, that's fascinating stuff. Can you hear me okay?
Yes, we can.
Okay, I'm just curious, you mentioned at the end that you're trying to combine the lipid nanoparticle technology. Obviously, you can encapsulate these structures very easily in LNP systems. What kind of objectives are you anticipating there?
Okay, so multiple objectives. This is very exploratory. So what we're going to try to do is, first of all, generate our spherical nucleic acids, and as you said, try to encapsulate them in lipid nanoparticles and see if the stability of the lipid nanoparticle, its ability to deliver to the cytosol, is enhanced using because ours are very simple modification, just a few alkyl chains on the end of a DNA or an siRNA. So that's one thing that we'd like to do. We have another technology, unfortunately, I didn't have time to talk about, whereby we actually take a poly-A system, and we have discovered a way to make poly-A, polyadenine chains, into fibers and into hydrogels. And so the exciting thing about this is that mRNA, of course, is polyadenylated, right? And so we would like to also use our technology, combine it with lipid nanoparticles, again, to see if that way we can actually enhance the delivery using this strategy for mRNA type delivery. We have two more technologies if you're interested. Would you like me to continue?
Well, yeah, I'm just curious. Because obviously with the lipid nanoparticle, you're going to get probably much better intracellular delivery than you might get with the, the unmodified DNA-based structures. And so, you might be able to see, you won't have to concentrate on getting out of the endosome so much as the subsequent events that you want to modulate. So, which could be an advantage. I mean, you're trying to build a lot of different things into the structure here. Sometimes it's easier if you only have one or two things that you need to build in. But anyway, that's
a very good point. Yeah, absolutely, a very good point. One thing that one thing that I dream about is the lipid nanoparticle technology is very mature and very established and has been tested so much. So what we really would like to do is to enhance it with our tools. And so I did talk about, the core of the lipid nanoparticle. We would like to also decorate lipid nanoparticles with different patterns, DNA patterns, DNA patterned presentation of targeting ligands. Targeting is very important, and so far, I don't know, maybe I'm wrong, but so far we don't have an FDA approved nanoparticle with targeting ligands on it. Peter, you might want to correct me on that.
No, that's totally true. It's because it's extraordinarily difficult.
yes, and so we have just fundamental questions that we'd like to ask. If we pattern our ligands on the exterior of lipid nanoparticles, how does that actually change how they are recognized? And does that make a difference? Because, right now, all we can do is isotropically just fill the outside, the shell, right, with the ligands. But maybe that's not as good. Maybe we need to actually pattern them in different ways so that they can be recognized multivalently or recognized by multiple receptors, let's say, on cells. So working on the outside of the lipid nanoparticles and on the inside, and seeing if our technologies can enhance these lipid nanoparticles.
Okay, that's great. Thanks very much for the lecture, the seminar. It was terrific. I think I'll let somebody else ask some more questions then.
Nancy Ford has given you a thumbs up. She said, "Fantastic talk, Hanadi." And then we have a couple of questions. So Marshall, maybe I'll get you to take the second one. But the first one says, "Thank you for the amazing talk." And the question is it possible to, instead of cage a drug, to cage a single cell? For example, for immunotherapy with cells that can fight cancer.
That would be fantastic. Yes, I don't see why not. In fact, one of our contributions a while back is we worked with models of cellular membranes, and we showed how we can take our cages and we can place cholesterols at specific positions, and those cholesterols anchor our cages on the exterior of a membrane, and therefore pattern ligands on the exterior of membranes. So yes, that is possible. Be great to actually apply some of these to CAR T therapies, for example. Open to any collaborations with that.
Great, thanks. Marshall, do you want to take the next question?
Sure. Actually, one came in through the chat from Terry Allen. Terry, if you want to ask it directly, go ahead. Otherwise, I'll read your note.
Yeah, well, I started out by saying very exciting and interesting. And then, of course, when I wrote the question in the chat, it was before you started to get into the point on your lecture where you were answering my question. But basically, it has to do with the in vivo characteristics of these structures: circulation half-lives, in vivo targetability, susceptibility to enzymatic breakdown, biodegradability, all of those things, that huge complex of pharmacokinetics and biodistribution that we'd like to know about, which are going to be critical to any successful application. I mean, you've made amazing progress so far. But in terms of in vivo applicability, there is much still to be done.
Absolutely. I completely agree with you, Terry. And we're going to be busy in the next few years. One thing that I wanted to mention here is I'm grateful for your network. I'm grateful for all the networks that actually bring together people like myself who are chemists and DNA nanotechnologists. So we're not actually experts in this, and people like yourself and others in your network, and Peter, experts. And I really would love to have more conversations and definitely collaborations on this. We are starting some collaborations with many of the clinicians in our university. McGill has a large number of excellent clinicians, but more collaborations and more in vivo models. That's really our what we lack in chemistry, right? Is this easy access to in vivo models, and that would be great to collaborate with people who do have that.
And thank you. At this point in time, I think it's important to try to focus on proof of principle in only one or maybe two in vivo models, and not spread yourself too thin. Mm-hmm. By involving too many collaborators, each of whom have their own particular models and particular interests, and getting quickly to that proof of principle for an in vivo application, I think might be something to concentrate on.
Thank you for the advice. That's excellent advice. And yes, absolutely. And if you have any other advice on where to seek that one collaboration where they will help us cross, let's say, the bridge for the SNAs, which are very simple to make. One thing I wanted to say is that we are collaborating with Alnylam Pharmaceuticals, and what they are trying to do is to see if their GalNAc structures can be conjugated to our sequence-controlled polymers, and whether we can actually improve the biodistribution and the pharmacokinetics of their structures. So we are going to start getting some of these answers soon, but yes, you're absolutely right. We need kind of one model and to do all of the studies so that we answer those questions.
Excellent. Well, the next question comes from Mohammed Al-Fach Salem, and Mohammed says, amazing presentation, and to your recent comment, he'd be excited to collaborate if there's any chance. The question is, for small molecule delivery, is it worth it to use the DNA structure, or is most of the potential relevant to the nucleic acid therapeutics due to the delivery challenges?
You're absolutely right, Mohammed. It's much more the nucleic acid therapeutic challenge that is important. I don't think that there's a lot of gain if you're not trying to deliver a nucleic acid therapeutic to use our structures. But what they do as well is they allow you to have combination therapies, right? Because their exterior is let's say gene silencing, their interior encapsulates small molecule drugs. Now you can actually explore synergistic effects in combination therapeutics using these strategies. And that's exciting because, again, they're very easy to make. They're made from a single molecule and they're spontaneously assembled. So it might be interesting to have that synergy.
Dr. Sleiman, would you like to make any closing remarks today in terms of your research or opportunities for our trainees or other investigators to collaborate with you in the future?
Well, first of all, I'd love to collaborate with investigators, and I would love to hear from the experts in your network on how we can actually take our chemistry and develop it for drug delivery. So, open to any suggestion. I also wanted to say thank you so much for inviting me, and I am deeply honored that I'm in the Pieter Cullis Invitational Lecture. He's been a true inspiration for all of us, and thank you, Pieter, for everything that you've done and that you're doing.
Thanks, Hanadi.